For this lecture, I will introduce you to various laboratory mathematics techniques and solution preparations. So one of the things we talk about in the laboratory is significant figures. So sometimes it's a little confusing as to try to determine how many decimal places do you go when you're doing calculations. So using more digits than necessary to actually perform a calculation and then actually reporting those results it can be a disadvantage. Number one, it's time consuming to go out several decimal places when you don't need to. But number two, it also can be misleading because you may think that this means the more decimal places, the more accurate the value, but this is not always the case. So when it comes to determining how far to take those decimal places out, you've a lot of times got to look at what the manufacturer of the instrument for that particular analyte has already determined. So a significant figure is known to be reasonably reliable. Once that's determined, that's what you want to keep suit with, whatever significant figure is established for that particular analyte or that value range. So the digits in a number that are needed to express the precision of the measurement from which the number is derived, that is known as the significant figure. And so we need to take the accuracy of the least accurate measure, or the measurement with the least number of significant figures, or the least number of decimal places, and we need to use that as the accuracy of the final result. So the book does kind of give us an example. There's a couple examples in there. So for example, if your particular analyzer's value has been determined that it reports out to 1 decimal place, so let's just say, for example, your result for a particular test is 10.0. So any calculations that you do, you would always want to round up to that 10th decimal place. Another example would be, let's just say you did some calculations and you got some series of numbers that you're going to have to work with. 206.1, 7.56, and 0.8764. So with those digits, you can see that our very first measurement, the 206.1, has the least number of significant figures. So it only goes to a 10th, whereas the others go out even more. So if we were going to do some calculations with these values, we would only take them to 1 decimal place. So that means we would do 206.1, 7.6, we'll round up, which we'll talk about that here again shortly, and then 0.9, because we're rounding that 8 up to 9. And so those are the figures that we would use in our calculations once we've determined our significant figures. So most of you are probably familiar with how to round numbers. So the book does give a few examples, so we're just going to kind of talk about those a little bit. So your test results occasionally, your calculations or your result that your analyzer reports out may go beyond what's considered your significant figure, so it may go actually into some insignificant digits. So your decimal place could get extended way out, like 6+ decimal places. So what you want to do is go through a process of rounding those numbers once you've determined what your significant figure or significant value is, and then you'll round based off of that. So when the digit next to the last one to be retained, in other words, the significant figure that you're keeping, is less than 5, the last digit should be left unchanged. So your last significant digit. So let's just say the example that was given in the book, 4 decimal places is considered your significant figure. So they actually gave an example of 2.13463. So if our significant figures only goes to 4 decimal places, then that 3 is what's going to need to be dropped. So we're leaving the 6. So we need to figure out, does that 6 go up? Does it go down? What exactly happens? So our 3 is less than 5, so we would actually leave that 6 unchanged. So our value would actually be 2.1346. All right, and then our next example that is given, the value is 5.34659. So with this digit, if we were just going 4 decimal places, the last digit that we're keeping, which will be that 4th decimal place, the 5, would need to be rounded up, simply because our next digit is a 9, and so we would round this up, and we would come up with 5.346. So that 5 would go round up to a 6. If the additional digit is a 5, so if that last digit is a 5, the last digit reported is changed to the nearest even number. Now this one's a little bit confusing because we've been talking about significant figures. So our examples given are 23.5 and 24.5. So with the 23.5, our last digit is a 5. So because it says that it's a 5, the last digit is reported is changed to the nearest even number. So when we talk about even numbers, we talk about 2, 4, 6, 8, or 0. And so 23.5 would go to the nearest even number, where our nearest even number to 23 is either 22 or 24. Well, we're obviously not going to round down, so it would go up. So this one would actually go to 24. And then our 24.5, our even number is 24. So the significant figures in rounding these values would both be 24. In the laboratory, we do see a lot of exponents. You got to remember that a lot of our values, especially when it comes to cells, there are thousands, hundreds of thousands of cells. So in order to kind of condense those numbers down a little bit, we do use a lot of exponents. And so exponents are used to indicate the number must be multiplied by itself as many times as indicated by that exponent. So our exponents often referred to as a power, or if you remember back from a high school math, raised to the 9th power, or the 9th exponent. And then of course our number that we're actually going to multiply would be that base number. So that's your main significant number raised to the 9th power, so 13th to the 9th power. The exponent is written as a small superscript figure that's up to the right, so you can kind of see an example of that here. Now, when will you actually see this in the laboratory? Well, we see this a lot with, like I said, reporting of cells. So, for example, if our patient's platelet count is 250,000, then how you would actually see it come out on a report would be 250 times 10 to the 3rd. So there's a couple ways you could report it, 250,000 or 250. So we usually refer to it in the lab. If we say this patient's platelet is 250, then we kind of all know that that's times 10 to the 3rd. But let's just say, for example, our patient's result comes out at 2.5 for their platelet value times 10 to the 3rd. Well, we know this is a critical value because if we were to translate that out, it would actually come to, and this is a point, 2.5. So if we were to move that decimal 3 places, so if this was our decimal place, it actually goes 3 places. So Three places would actually be 2,500, which is an extremely low platelet count. And so that's why it's very important to understand and get used to the various values and how they're reported as you're working through your coursework, so that you could understand the difference between what could be considered normal, 250,000 or 250 times 10 to the third for a platelet count, or 2,500, 2.5 times 10 to the third. So there's a significant difference between being critical and normal if you don't understand how those exponents work. So the density of something is the amount of matter per unit volume of a substance. And so all substances have this particular property, not solutions only. And so an example of this would be expressing density in the form of specific gravity of a substance. So specific gravity, you've probably heard this already from your analysis, is the weight of 1 ml of a solution compared with the weight of 1 ml of pure water at 4 degrees Celsius. And so 4 degrees Celsius is refrigerator temperature. And so specific gravity is used a lot in clinical or research laboratory when there's any sort of preparing of a dilution from something that's concentrated like an acid. So when you might actually use this will be when you're trying to actually determine how to prepare some different solutions. So the specific gravity multiplied by the percent assay will equal your grams per ml for that particular compound. So there is an example given in which the hydrochloric acid has a specific gravity of 1.25 grams per ml, and then that assay value of 38%. So then you would determine what is the amount of hydrochloric acid per ml. And so if you were to work out that equation, you would get 0.475 grams of hydrochloric per ml. And so once you've actually determined the grams of hydrochloric acid, then you could actually go further and do calculations if you were trying to make a solution and determine exactly how many mLs of acid you would need to actually prepare a particular concentration of that hydrochloric solution. So if you have a laboratory procedure that calls for a 4% hydrochloric acid solution, then you would be able to take your powdered solution or your concentrated solution that you had, do these calculations, determine exactly how much you would need. If your procedure called for 20 mLs of a 4% solution, then you could do the calculations to determine exactly how much you would need to prepare that. And so this is where you would actually use these figures. So solutions are just combinations or mixtures of different substances. So if you're making up a solution, it's going to involve two things. You have the substance that is being dissolved, or the solute, and then the substance to which you are dissolving it in, or the solvent. With working solutions, it's necessary to know or be able to measure the relative amounts of substance that's in a solution, because this is going to give you the concentration of that solution. So just like the previous example, a procedure might call for a percentage of a solution. It may call for a specific thing that you have to determine exactly if you have the correct concentration. And so the concentration is the amount of one substance relative to the amount of the other substance in that particular solution. So like I said, if it's calling for 4%, then 4% of that solution, that total 20 mLs, 4% of that is your actual hydrochloric acid. So it is, when you're expressing it with a series of weight, the weight's going to be the mass of it per unit volume. So this is something that's going to be expressed in like grams per ml or milligrams per ml or milliliters, or it could be grams per liter. So you're going to have a mass with a volume. Then you also, it could potentially be expressed as a volume per volume. So mLs per liters. A percentage is going to be a parts per the 100. So like I said, 4%, a 4% solution. And then you have molality versus molarity, osmolality versus osmolarity, which we will discuss in a little bit more detail. And then you also have titers, which is basically reduced concentrations of something as it is diluted out. So we'll talk about that a little bit further as well. When referring to molality, that is the number of moles of solute per kilogram of solvent. Now, molarity is the gram of molecular mass or the weight of a compound per liter. So you're kind of looking at a weight versus something in a volume. And so when we talk about the molarity of something, this is the weight per unit of volume. And so basically we're using this to express a method of concentration. So the concentration of something, the molarity of something. Another way to define it is the number of moles per liter. So again, you've got something that's in a solution or a volume of something. And so a mole is the molecular weight of a compound in grams. So 1 mole is equal to 1 gram of molecular weight. The number of moles of a compound equals the number of grams divided by the gram molecular weight of that compound. And so basically what you're doing with these things is it's going to kind of help you to determine the percentage of something within a solution. And it could be, you're not really going to use this a lot in the clinical laboratory setting, but if you're in a position of maybe some sort of research, then it may be times that you might need to do some calculations like this to figure out how to calculate how much you would need of one solution to balance out another solution. So if you're trying to make an acid not as acidic anymore, then there may be some things that you would need to add to it to balance out that solution. So this is where you're getting into some of those calculations. Osmolality and osmolarity are, when we look at those at the clinical laboratory standpoint, they're basically going to be used to evaluate the blood and the urine for that balance and that osmotic pressure is what we're looking for to make sure that the body is able to hold that equilibrium throughout your organ systems. And so osmol as a unit of measurement describes the number of moles of a compound, so maybe sodium or glucose, that is contributing to the osmotic pressure of a chemical solution. So in order to maintain that balance within the body, your kidneys need to be able to accommodate for any kind of influxes or drops in any of those particular electrolytes. So this is where determining the osmolality or osmolarity comes into play. So we do often use proportions and ratios in the clinical laboratory setting. Proportions are used to determine a quantity from a given ratio. So the ratio itself is expressed as an amount of something compared to an amount of something else. So ratios are described as a relative amount, and you have at least two values that are going to be involved. So for example, 5 grams of hydrochloric acid is dissolved in 100 mLs of deionized water. So it's basically something else that is dissolved into something else. So we have the 5 to 100. If you were to write that out in a decimal, it would be, you're going not the tens place, but the hundredths place. So you would do 0.05. So you have 5 grams of hydrochloric acid dissolved in 100 mLs of deionized water. In setting up a ratio and proportion problems, the two ratios being compared must be written in the same order. So they must be in the same units. So if you were doing a problem where you're talking about 5 grams of hydrochloric acid dissolved in 100 mLs of deionized water, then if you were trying to figure out a concentration of another one, so if you knew that it was 1 gram of hydrochloric acid dissolved in 10 milliliters of deionized water, then you would express it the same way. What's being dissolved into what it is dissolved in. So that's how you would write out those ratios. You also need to kind of understand the concentration of a solution. So we will use this a lot working in the clinical laboratory. To relate different concentrations of solutions that contain the same amount of substance or a solute, you have a basic relationship or a ratio that is used. So just kind of explain to you what a ratio is, and then you have to use that to kind of help you determine concentrations of things. So the volume of one solution, we're going to call that volume 1, times the concentration of that solution, so concentration is going to be expressed as C1, equals the volume of the second solution, volume 2, times the concentration of the second solution. So with either one of these, even though your volumes might be different for each solution, your concentration of that solution should be the same. If any of the 3 values are known, the 4th must be determined. So usually you're giving what the concentration, the volume and concentration of something is. So you may be using this equation solution where you're trying to dilute out something to determine exactly how much you would need to get the concentration you're looking for. And so you will use these occasionally, this particular equation a lot. So you do want to be familiar with it. It's a fairly simple equation to memorize too. The volume times the concentration of one substance equals the volume and concentration of another substance. You will also perform dilutions when you're working in the clinical lab, and we will actually do a lab exercise where you are working on dilutions and understanding what dilutions are. It is often necessary to make dilutions when you're analyzing something to kind of make it a weaker solution. And so cases like this might be when you have values that when you're doing a test and those values go beyond what your analytical measurement range is for that analyzer. So you might have to do a dilution to drop that figure down so that you can get a value that is able to be read on the machine. But once you do that, you've also got to remember you've diluted that patient's sample, so now you need to make accommodations for that. So if you've diluted it out, then you need to adjust it by that dilution factor. So if it's a single dilution, it's pretty easy to do. Like if you just do a 1-to-1 or you diluted it one time, 1 part of your patient sample with whatever you're diluting it with, and then you would work that out. So we are going to work on this right much in intro, and then you will see it again throughout the program, how to determine dilutions, because this is something that you're going to do quite often in your career. While some of the machines will do the dilutions and calculations for you, there are still some times where you might have to manually do a dilution. So when we talk about dilutions, we talk about the sample volume, so how much you use. So maybe you use 1 ml over the sample volume plus the diluent volume. So if your sample or your dilution that you're making calls for us to use saline, if you use 1 ml a patient sample, then your total volume would be your sample volume, so that was 1 ml, And let's just say we're going to use also 1 ml of saline. And so what our dilution is going to come out to is going to be 1 ml over our total volume, which is 2 mLs. A serial dilution is an actual set or a series of dilutions in which each time that you perform the dilution, you're cutting that patient sample by whatever dilution factor you're using, or it's based off of that volume. So in this particular example, you take one specific volume of your patient sample and then you dilute that in half. Then you take that volume, dilute that volume in half. So you keep going in the series of test tubes where you're actually diluting it by half each time that you do it. So if your result was originally 100, let's just say we were diluting a glucose, the original result was 100. So when we dilute it in this first tube, we've got 1 part patient sample, which is 1 ml, to 2 parts whole. So our 2 parts whole would be 1 part of that's going to be our patient sample, and 1 part of that is going to be whatever we're diluting it. So in this case, if it was saline, then half of it is patient and half of it is saline. Now granted, it's all mixed up, but obviously that result would need to be multiplied by 2. So that is our dilution factor. So that last little digit there, that 2, is our dilution factor. But you still got to keep in mind, out of those 2 parts, only one of that is actual patient sample. So half is patient sample. So obviously whatever result that you get should be half of the original. So in this particular case, we would expect to get a result somewhere around 50. Now, if we take 1 ml of this that's already halved in this first tube, tube number 1, and we put it into the second tube that has 1 ml of solution, then we're cutting that half and half again. So 25. However, you see that 1 part is our patient sample, and then the other 3 parts are our dilution, our diluent, in this case would be our saline. So if we were to multiply 25 by our dilution factor of 4, or times 4, then that's going to give us 100. So you can see as you go, this value of our patient sample, the original part of the patient sample, is going to get diluted each time. And you can see as we do equal parts, 1 ml of our diluent or our saline to the 1 ml part of patient sample, which you can see it's been diluted each time. As we move that forward, it's going to dilute out every time, so our dilution factor is actually going to double. If we had used different amounts, this could, be altered a little bit. Let's just say we used 1 part in that first tube if we used 1 ml of patient serum, but we used 2 mLs of our our diluent or our saline, then we would actually have a 1 to 3, because 1 part of it is patient, whereas 2 part of it is diluent. So that would change up those values at the bottom. There is some laboratory exercises, online laboratory exercises, where you will practice this, but we will also practice it in our hands-on skills campus lab as well. The standard solution is one that contains a known or exact amount of substance that's being measured. So for example, if you know that you're measuring glucose in a particular solution, the value of that is already known. We know how much is in that solution. And so we use these a lot for working standards, which are prepared from stock solutions. And so we use these a lot in spectrophotometry, where we have a standard. We know how much is in it, so that we can use that to assess how exactly we're using that measurement in our spectrophotometry lab. So it kind of gives us our set point value. We also use blank solutions which contain the reagent, but it's nothing else in it. No substance is in there. So this allows us to account for any kind of color variations or anything in that reagent. And so it allows us to measure only the substance and not have any interference from the reagent itself.